Good afternoon. My name is Patricia Mirol. I'm a researcher at the National Research Council in Argentina, and my group works on conservation genetics of mammals, endangered mammals, at the Museum of Natural Science in Buenos Aires. Just to put you in context, Argentina represents the southern edge of the distribution of many species. This has consequences. Because habitat loss and fragmentation frequently has a peripheral center pattern, the edge populations are the first to retract and the first to fragment. This means that these populations are going to be isolated, are going to have small population sizes, frequent gene drift, low gene flow, and low genetic variability. However, these populations, as damaged as they could seem, are very important from the point of view of conservation. Why? Because they can maintain unique genetic and phenotypic characteristics. If you have a look at the Argentinian Red List of mammals, you will find that 33% of the species fall within some category of risk. But it is important to have in mind that local and global categorizations are not always the same. And I'm going to show you some examples of this. These are some of the mammals we are working with in our group. And I think the perfect example to show you the differences between categorizations is the jaguar. The jaguar in Argentina only survives in 5% of the historical range of the species in the country. However, because of the distribution in the rest of South America and in Central America, IUCN has categorized the species as near threatened, while the Red List in Argentina has categorized the jaguar in Argentina as critically endangered. Similar cases are the white-lipped peccary and the giant armadillo. I like to talk in this presentation about how we are going, how we are transitioning from conservation genetics, that was at the beginning of our work, to conservation genomics, and what are the issues we are finding along the way. So to start, I will explain our first works that were with the jaguar. We started approximately 10 years ago. As you can see in this map, there's approximately 200 jaguars in total in Argentina, which are distributed in 3 ecoregions: Yungas at the west, Chaco region, and the Atlantic forest that continues in Brazil. We collected approximately 300 samples, 90% of them were feces, and using 10 microsatellites, we were able to identify 54 individuals. We were working with traditional molecular methods. These 54 individuals were grouped in 2 different genetic units. On one side, the Atlantic forest unit, very different from the second unit that was formed by the Yungas and Chaco. What it was common of these 2 genetic units was that we could detect signs of inbreeding and low effective population size. If you look at the whole distribution of the species from Mexico to Argentina, you can see that the genetic variability that we found in Argentina is among the lowest in the whole populations studied. And it coincides with most of the variability at the peripheral populations, which is always, they are always the lowest ones. So we propose different management actions, but 2 of the main of these actions were, by one hand, the restoration of habitat connectivity between Yungas and Chaco, as you can see in this map. Why? Because even when we found that these 2 regions form a unique genetic cluster, we know that ecological connectivity has been lost during the last decades. So it is very important to reconnect these two regions so they do not become isolated. And the second recommendation that is very important is the preservation of unique and rare alleles, because Chaco is the region with the biggest habitat for the jaguar. However, it has the fewer animals. And these animals have very unique genetic characteristics. So we have to manage them in a way that we can preserve the genetic characteristics of these animals. So going now to my second example, the marsh deer. As you can see in this map here, most of the historical distribution of the marsh deer in South America is now extinct. It survives in very few populations, isolated and fragmented. In Argentina, we have 4 of these fragments, 3 in the north of the country, and 1 quite south, in the very southernmost part of the distribution, that is at the end of the Paraná River between Buenos Aires and Entre Rios. In this case, we wanted to move to NGS. We were wondering between using SNPs or microsatellites, but because we don't have the genome available for the species, and again, because 90% of our samples were feces, we decided to go for amplicon sequencing. We could do this because we work in collaboration with 2 groups. One group in Brazil, where we did the partial genome sequencing, and a second group in Canada, where we did the amplicon sequencing. I will show you some of the results. We were able to obtain complete mitogenomes from one individual from Brazil and another mitogenome from an individual from Argentina. And using 2,000 base pairs of this mitochondrial DNA, we were able to construct this network where you can see that there are different mitochondrial lineages. All the reds here correspond to individuals and haplotypes from the center of Brazil. The green here are the populations in the north of Argentina, and the very important yellow here, very distant from all the rest, is the only and unique haplotype we found in all the animals, approximately 30, that we examined at the southernmost population. So we decided to have a closer look at this population. And what we found, this is the southern population, is that it's consisted of 3 different fragments which have the dynamics of a metapopulation, in which there is 1 fragment connected with the other 2 that is working as a connector, while there is no significant migration or movement of animals between the 2 fragments at the south. We were also able to calculate to estimate the effective population size, which was below 50, and this made us propose to change the status of the species, at least of this population, from vulnerable to endangered, because less than 50 individuals implies that the population is endangered. Now, we are starting a new project of whole genome sequencing. This project is supported by funding by the Ministry of Environment of Argentina and the National Park Administration. The idea is to do the whole genome sequencing of some endangered mammals from the north of Argentina, and also do afterwards amplicon sequencing to study variability, because for some of these animals, there's no knowledge of the genetic variability in our country at all. So now I would like to come back to the title of my presentation, From Conservation Genetics to Conservation Genomics in Argentina, when science is not the issue. We know we have the technology, we have the knowledge, yes? But here, look at this slide that I'm showing you. This is more or less the amount of money I calculated needed for each one of the 3 cases I showed you before. And it goes from $13,000 approximately to $29,000 approximately. And the truth is there is no grant in Argentina that is able to pay for the expenses of these consumables needed to do the whole genome sequencing and amplicon sequencing. And unfortunately, I think this is the case for most of the Latin American countries. And I think this is why even when we are a hotspot for biodiversity, there are so few genomes of endangered animals in this part of the world. So thank you very much for your attention. Muchas gracias.